All right, and next up we have Dr. Perry from Cook Children's Medical Center, who is going to give us an overview of clinical trials in Dravet syndrome.
All right, you are asking yourself right now, Scott, who did you piss off to give you the spot right before lunch? I don't know. I think I'm a nice guy, but apparently somebody's angry. You know, I'm going to try my best, because rest assured, people, I am also hungry. Okay, so we're going to move on. All right, so I just want to gauge a little bit and understand where I'm at here. So just raise your hand if you are the caregiver of a person with Dravet syndrome in the audience. Okay, lots and lots of you. Now, keep your hand up if you are a caregiver of a person with Dravet syndrome and you have participated in a clinical trial. Okay, so a good number of you, but some of you have not, all right? And then who in here is with the industry? I recognize many of you, but just raise your hand, the industry folks are here. Okay, so I tried that when I was given the topic, I was asked to give an update on clinical trials. And I could go over a lot of clinical trial data, but you had a whole day of looking at clinical trial data yesterday. So I am going to touch on that a little bit, but that's not really what the talk is about. So really what I want to do so there's some disclosures. I work with lots and lots and lots of people. All right, so here are the goals. So what I want to do is really review kind of the basic concepts of clinical trial design and research participation, because for those of you who have never been in a research trial, I want to talk to you about why you ought to think about it and why it may be worthwhile. As part of that, we'll talk about how clinical trials are designed, some of our common clinical trial endpoints. Then we'll talk about some important things to communicate to families and people who participate in research, maybe some things that people don't always think about, and this is also for those industry folks in the room who can think about some of the struggles that we have, both the caregivers and the people conducting the trial when we deal with these. And then finally, at the end, I will rapidly go through some of the data for what's out there, what's new, what's coming. So if you missed yesterday, you'll get a kind of a quick overview, okay? So who should participate in a clinical trial? Absolutely anyone should participate in a clinical trial. It is worth everyone thinking about clinical trials. When you go and speak to your provider, your physician, about what they can do to help you, you need all of the options on the table so you can decide. And those options range from medicines to surgeries to data to neuromodulation to research trials, et cetera. So anyone should consider research trials. When you're doing it, you just need to consider, like, does it meet your current goals? Does it meet what you're trying to achieve? And there's lots of things you would want to think about. What kind of trial is it? Is this an open label trial where you absolutely know you're going to get the drug that is being studied? Is it a double-blind placebo-controlled trial where you may get the active drug or you may get the placebo? And if you do, how long are you going to be in the trial? Maybe you want to know, like, what stage are we in? Is this a very early stage of the trial where we know some safety data, but that's really what we're trying to gauge is safety data, versus a later trial where we've already had pre-existing trials, we understand the safety, we understand the dosing we're supposed to use, now we're just trying to prove it works. And again, you know, what are the risks and the benefits to you? What are the risks and benefits to the person you care for and you're making decisions for? So there's various stages of clinical trial design. This is just a very basic kind of high-level understanding of that, all right? So phase ones are usually we've got a drug, it's been studied in animals, it's been studied in the lab. Now we're giving it to healthy people just to understand how is the drug metabolized, how does it work in the human body, and is it safe? In phase two, usually that is being studied in a very small group, but a small group that has the disease in question. So these are not going to be really big trials. These are much smaller, studying for us, you know, kids or adults with Dravet syndrome, and we want to understand better now in this disease, how is it safe, and somewhat is it efficacious? Does it work? But mostly, is it safe? What doses are we going to use, et cetera? Once we get that data, then that's the phase three. Phase three are the big trials that have hundreds of people in them that are placebo-controlled, double-blinded, just to make sure we're all on the same page. Placebo-controlled, again, means that some people get the active drug, some people get a placebo that is not active. They are double-blinded, meaning that I, as the physician, do not know what you're receiving, and you, as the patient or caregiver, do not know what you're receiving. And they're randomized, meaning, when you come in, we have a system that decides which of those groups you go in. And it's not, predetermined. And the reason for that is to get rid of bias as much as possible. Because you can imagine if you've heard about a drug and you hear that drug really, really works well, and then you come get in a trial where you're getting that drug, you have a higher likelihood to believe things are going really well. And as a result, that hurts the end result of the data that we get. And then finally, phase 4. Phase 4 is really post-marketing. The drug has been approved, it's on the market, it's being used, and now we continue to study how it's being used, how it works, what its safety is. The reason for that is because, as I'll talk about in a minute, clinical trials are very regimented procedures. Everything we do in a clinical trial is laid out in the protocol, and that is not real life. So when you get into real life, real things start to happen. You're starting to use other drugs that weren't allowed in the protocol. You may be using it slightly different ways, things like that. And so we need to continue to follow it long term to make sure that the data we got from the phase 3 is actually accurate. So let's talk about how we start out in a clinical trial. If you decide you want to do a clinical trial, the first and most important thing that you're going to do in the clinical trial is give informed consent. Informed consent is a very meaningful conversation between you and the people conducting the study that allows you time to understand exactly what you're signing up for and to make sure that you have asked all the questions that you need to ask to make sure it's right for you. So there are essential elements to informed consents that you should see when you are going into a trial. These are things like making sure you understand this is research. You're going to be part of research. There are risks. There are potential risks, there are potential discomforts to the treatment. There are also benefits. There are also alternatives. You don't have to do what's in the research trial. There are other options that you can do, and there are risks and benefits to those options. How are we going to hold your data? How confidential is it going to be? Who are we going to share it with? What happens if during the trial something happens to you and you get sick, maybe because of the trial itself? Who's going to take care of that? Is there compensation for that? Who do you contact when you have questions? And most importantly, making sure you understand this is absolutely voluntary. At any moment, you can stop being in the trial. You are not required to stay if you don't want to be. So all of those things need to be discussed with you. You need to understand that before you sign on to do it. Once you've signed on to do it, and then you're in, and we are dealing with a protocol. And as I said, these protocols are incredibly wordy. They are hundreds of pages of stuff, hundreds of pages of how we're going to do absolutely everything in the trial so that every place that's conducting the trial is conducting it exactly the same way so that we have very, very solid data. Everyone that participates in the trial at any one site has been trained on the protocol. They are all delegated certain jobs that they must do. The data that we collect is monitored for accuracy, so people will come to our sites, they will look at our data, they will make sure our data matches with the source, matches with what's being collected. So very regimented, and I say that just because in a few slides I'm going to talk to you about, for those of you who have been in clinical trials, some of the things you might experience, and hopefully it makes a little sense of why we put you through all the questions and things we do. Adverse events, as I said, after informed consent, patient safety. Patient safety is really the most important thing about research. We need these studies to be as safe as absolutely possible, and so we collect adverse events throughout research protocols, and we collect absolutely everything, regardless of whether it is related to the treatment under study, right? So once you enter a trial, if you then went home and slipped and broke your foot, that is an adverse effect. It may not be an adverse effect of the drug, but it's still an adverse effect. It must be recorded, and then myself or whoever is the principal investigator has to determine, do we think that adverse effect was the result of the drug, or do we think it was unrelated? So we will record anything that happens. Serious adverse effects, obviously, are very important. Serious adverse effects are things like death, which is obviously serious, hospitalizations or prolongation of hospitalizations, or other things that really significantly disrupt daily life. And then these are kind of the common steps of a research trial. So once you've consented, you've entered the trial, you're usually in a screening and baseline phase. During the screening and baseline phase, this is when there's going to be a lot of testing. There's going to be lab, EKG, maybe there's some scales done. And that's all to make sure that you meet the inclusion and exclusion criteria, because there's going to be a long list of things we have to say that you're good for the study or you're not going to be allowed to be in the study. And so during the screening phase, we do that. Plus, that's also for Dravet and most epilepsy trials, that's the time where we're getting the baseline seizure frequency. So during that month, it's usually about 28 days. You're not changing any other treatments. We just need to know how are you on a usual day with nothing being changed. And that gives us our baseline frequency of seizures. Then you enter the treatment phase. The treatment phase, on average, lasts 12 to 16 weeks in most studies. So this is the time where you'll be given the investigational treatment or you'll be given the placebo if it's a double-blinded trial, for example. And then after that, then you enter the end of study or possibly an open label extension. So many trials will have, because people get enrolled at different times, you get to the end of your 16 weeks, you have the option to discontinue the treatment and wean off of it, or you can enter an open label extension. When you enter the open label extension, that means you're getting into a different trial using the same drug, but you're actually getting the drug at that point. So if you were on placebo, you'll be switched over to the actual drug. If you were on actual drug, you would continue on it. You still won't know what you were on during the study, mind you. And then the open label extension generally goes on until the phase 3 trial has been completed and the FDA has reviewed the data. And if the drug is approved, then great, you get switched over to the commercial product. If the trial was a failure, then we talk about whether or not you should continue the drug or whether we should be weaning off. Okay, let's talk about outcomes in clinical trials. Now, what do we collect? Here's the key takeaways kind of from this section. So in general, clinical trials for epilepsy focus on seizures as the primary endpoint. Makes sense, right? Secondary endpoints are generally non-seizure outcomes, so maybe behavior, maybe sleep, maybe developmental scales, things like that. In general, trials, especially for drugs, are designed to demonstrate the absence of worsening of things. So the trial is designed to show that the drug doesn't make your behavior worse, it doesn't make your sleep worse, things like that, but they're rarely designed to show us that those things are better. They're really only focused most of the time on showing us that the seizures are better, and those other things the study's just not powered for it. They don't have enough people in the study oftentimes. Also, the other problem we have with non-seizure outcomes is a lot of the scales we used were not designed for people with developmental and epileptic encephalopathies like Dravet syndrome. And so for any of you who've been in the trial and had done some of these scales that we give you, sometimes get frustrated with them because they ask questions that you think are ridiculous. You're like, my kid absolutely can't do this, so why is this even a question? Like, why? And it's frustrating because the scales aren't designed for these kids oftentimes. So then the next factor to talk about is, since we're counting seizures, usually, is how do we determine that? So again, for those of you who have been through clinical trials, you probably went in and you sat down with a person like me, and I asked you an excruciating list of questions about your seizures. And you came in and you said, oh yeah, my kid has focal seizures and they have absence seizures. And then I said, okay, I'm going to stop you, and now you tell me, what does it mean to have a focal seizure? Exactly what does that look like to you? Because then when you described the event to me, I was like, actually, that's not a focal seizure, that's a different type of seizure, right? So we ask you all of these questions to understand what you see at home, and then we assign it the name that we think is appropriate. Even we don't get to make the final decision because it often goes to a third-party consortium who then looks at it and says, yes, we agree with you, you have labeled the seizure the correct thing. Sometimes they send it back to us and it's like, are you sure that's the type of seizure it is? Maybe it should be called this instead. There are multiple seizure types, right? Some are easy to deal with, some are not so easy to deal with, at least from a research trial standpoint. So, in general, it's pretty easy to count generalized tonic-clonic seizures, grand mal type seizures, right? It becomes increasingly harder to calculate others. For tonic seizures, if any of you, have kids with tonic seizures, those happen a lot at night, they happen a lot of sleep, so you miss a lot of those. Absence seizures, very short staring episodes, or myoclonic seizures, these little quick jerks, you can't count that. There'll be hundreds a day. You can't keep up with that. You can't walk around behind your kid clicking the clicker all day long. That's just not gonna work. And so that's why things like absence seizures and myoclonic seizures are rarely ever included in clinical trials, because they're just not reliably countable. Oftentimes in clinical trials, you'll see that they're designed with what is really a group of seizures. So many studies will study convulsive seizures, or what they call major motor seizures, and that includes multiple seizure types. We also see drop seizures mentioned a lot in trials, and drop seizures also are multiple types of seizures. It's useful information. It's a good way to kind of group everything together. As a physician, when we deal with those studies, we also like to look at the data and understand, okay, within the drop seizures, what was the response of the individual seizures, because that's quite informative for us in treatment. So I talked a little bit about characterizing the seizures. So you go, and there's like 3 main steps in characterizing seizures. One is caretaker description, okay? This is you giving us a verbal description of what you see at home with as many details as we can get from you. If you have video of the event, that is so helpful because we can really get to where we need to get to a lot faster. The next piece of that is physician characterization. So I take the information you give me, and then I determine, based on what you've told me, what type seizure I think it is, and that's what I put that you're going to count during the study. And then, as I said, oftentimes that information then goes to another group, often the Epilepsy Study Consortium, who is a group of kind of independent providers that then take that data. So they're not, they're kind of a third-party validity team, right? They look at what we submit and they say, yes, we agree with you, that's what that seizure should be called, and that will be counted, or they say, no, we have some questions, we need to clarify this seizure type to make sure we're counting the right thing. Then you go home and you count seizures. What can be more exciting than counting seizures for 4 months plus, right? And there's a few ways you can do it. There's one way, it's called paper and pen, all right? You go home with a piece of paper calendar, and every day you write down the types of seizures. You might write down whether you gave rescue medicines. You might write down whether it was a cluster. You write down the total number. There's some great things about paper diaries. They're super cheap. They're really easy to use, and if you make a mistake, you can put a line through it, initial it, date it, and put in the new data that's correct. That's awesome. The bad parts about it is the piece of paper does not call you to remind you that you need to collect the data, okay? It doesn't do that. It also is an extra step. So you bring us the paper, and then we still have to put the paper in the electronic format for it to go to our sponsor to review. Really easy to lose it, and of course the data is delayed until you come to the clinic and give it to us. We don't have the data. Oftentimes what happens, especially with paper diaries, is you get what's called parking lot data, which is that you came to your appointment with me for your research trial and you haven't been recording seizures like you're supposed to, so you record them all in the parking lot, and then you bring it in and you give it to me. And I say, curious that you had the exact same number of seizures every day for the last 14 days. But anyway, it does happen. So there's some downsides to paper diaries. Electronic diaries, usually some type of mobile device, it's like an iPad or a phone or something they'll give you. This does the same thing the paper diary does. It collects the type of seizures, how many you have. You put it all in the electronic format. And this can remind you. So each day, if any of you have done these trials where these things have alarms, you probably were annoyed by them because every day at a certain time it starts ringing and telling you that it's time to put in your data. And so you get it, you put your stuff in. So it's nice that it reminds you the data is accessible and uploadable, so we have it quickly. Downsides, for the sponsor, it's an increased cost. For you, it's usually IT, right? Technology never works exactly like we want it to. So you lose your password, or you put the wrong thing in, but you already hit submit. And when you put the wrong thing and hit submit, then you got to call my research nurse. My research nurse has to unlock it. She has to go in and try to change it. So it's kind of a pain. But there are some advantages. And then finally, there's something, an automated phone response. So basically, they'll either call you or you call the phone. It's like a phone tree. You basically are putting in numbers and codes for what type of seizure and how many. Those are good. Those can be tough to work with, though, because if you make wrong answers, again, backing up and correcting things is not always super easy with that approach. So what else is important to measure? So we talked about seizures, but as you all know, seizures is only one of the problems we have to deal with. And in Dravet syndrome, like many of the other developmental and epileptic encephalopathies, there are lots and lots of other comorbid conditions that we have to deal with. And Dravet syndrome has done one of these programs where caregivers and people that deal with Dravet syndrome are in the room with people from the FDA or in the room from industry folks and talked about like what is important to us to measure. So when you do your next clinical trial, what should you be looking at? And as you can see, there's lots of things that are important. Lots of other things happen in Dravet syndrome, and it's just a matter of how you measure them as to whether we could do it. But as we start to get to disease-modifying therapies, these other non-seizure things are going to be super important. This is just for LGS. LGS has done the same thing, showing multiple other factors that need to be evaluated. And this is just a good example. You may have seen this figure from other people's talks, but like all of the things that happen. You need to know, number one, all of the things that happen in Dravet syndrome, but in order for us to design these trials and understand if the drugs we're using actually are changing the course of your disease, we have to understand the course of the disease. So it's one thing to understand that all of those things listed in the table happen in a person with Dravet syndrome, but you also need to understand when they happen in a person with Dravet syndrome and what is their trajectory over time. So sure, you got developmental delay. How much developmental delay? How does that change over time? Because if we don't understand the course, then there's no way to compare to understand, does the treatment alter that course? So that's why we've done natural history studies that some of you may have participated in the past. These are just a few more scales that you'll often see us use. The Clinical Global Impression of Change is basically a kind of catch-all instrument where basically the caregiver has one, also providers have one. It's basically asking compared to, let's say, compared to prior to receiving treatment, is the overall condition of the patient, very much better, very much worse, and there's everything in between. It's just a scale that you pick a number. Then we have things like the quality of life in childhood. We have the Vineland Adaptive Behavioral Scales. The BRIEF is another survey that we use to measure executive function. But as I said, a lot of these scales just aren't quite adequate enough. So there is a push to try to develop scales that are more applicable to this population. There is something called the Inchstone Project, which is ongoing. It's started by the SC and 8A and a few other organizations have been part of that to really try to create measurements that are able to measure the inchstones of success, because their thought, and many of those developmental epileptic encephalopathies are very severe, and more severe than Dravet syndrome, if you can believe that. But when they're in trials, they say, hey, my kid is better. The scale you gave me doesn't measure that my kid is better because it measures really big changes, and my kid has made inchstones of changes, but it's still really important to me, and it's still meaningful. And so they're trying to develop scales that can measure that. There's also been some scales developed, this for CDKL5 deficiency disorder, specific to the disease. And this same process is now being done for Dravet syndrome. So DSF has funded Megan Abbott, I believe, at Colorado, right, to take this and really help create it more for Dravet syndrome. So we will have some scales that we're working on. Okay, a few other things to consider in clinical trials. There's things to manage. And some of these things I'm just telling you so you're aware of, and you try not to do them if you're in a clinical trial, but I can't stop you, I guess. But social media, keep it to yourself. You don't need to share with everyone you're in a clinical trial. You don't need to share with everyone how great or how badly it's going for you, because that biases the population. So as much as you can, it's great if we just keep it to ourselves. Then, we struggle. As I said, these can go on for a long time. If you're counting seizures every single day, you were in a 4-month trial, then you enter the open label extension, it's still going to be 2 more years before the drug gets approved, and we're going to ask you to keep counting seizures for 2 more years. It becomes hard, right? Obviously, that's where it becomes difficult to ensure that the data is quality all the time, because we're trying to encourage you to not do it in the parking lot. It's important when the trial starts, if you're in one, that you clarify all the do's and don'ts, and that whoever the investigator is clarifies that with your physician, right? Every medicine you get on, you get an earache or you get a cold or whatever, somebody puts you on the medicine, we have to know that you've been put on that, and sometimes those things aren't allowed, so it's always nice to be as clear as we can with your home physician what you need to do. It's also important for us as investigators to keep your primary physician informed along the way, whether that be your primary care provider or your neurologist, about how things are going. And then finally, I just want to talk just very briefly encouraging people to maintain their relationship with their primary neurologist. So this happens a lot. And I say this because I say breaking up is hard to do, okay? I see a lot of people that come to me for clinical trials, and they came to me from another neurologist. I want them to maintain the relationship with the neurologist they came from. Number one, because lots of them come to me from out of state. So if you get sick and you end up in the hospital, you're not going to end up in my hospital. You're going to end up in your local hospital. So they need to still be involved in your care, number one. Number two, once the trial's over, then we often get into a spot where people are like, well, can I continue coming here, or do I go back to my primary neurologist? I'm happy for you to continue coming to see me, but again, that may not always be the best thing for you to do. The other thing is, I think from a patient perspective, it's probably really hard to end a clinical trial because several things happen. Number one, your frequency of visits decrease. You've been seeing the neurologist, every month for the last, however many years of the trial, and then all of a sudden you're back to real life where it's like every 6 months or more, depending on the availability of your neurologist. Your access to the care team diminishes. When you're in a clinical trial with us, you've got my nurse Diana's text. You can text her any time of the day and get questions answered or whatnot. When the study's over, you don't have that anymore. Now you got to go to call, you got to put in a message, you got to wait for a response. So it's really different. And then it's back on you to deal with scheduling and making sure everything's coordinated. It's not part of a protocol anymore. And I know those things can be difficult. And then you go back to your home physician, and you can have some other difficulties too. Number one, your home physician wasn't part of the trial. So they may have no idea how to use the medicine. They haven't, many of you, for instance, for the Epidiolex or fenfluramine trials probably experienced that. You go back and they're like, I don't know how to prescribe that medicine. I'm not part of that. So, that's on us. We have to educate your providers how to do that. Okay, now, let's rapidly review what's available right now, okay? We're going to go through these quickly. All right, LP 352. I'm not a marketing guy, but I don't like that name, so we're going to have to get. It's not catchy. So this is Longboard's drug, bexicaserin. So this is a drug. Its mechanism of action is a 5-HT2C superagonist. So this is similar. Fenfluramine is a drug that acts on these HT receptors. The difference with this drug is it's very specific to the 2C. And the reason that's important is because when you have a drug that binds to multiple receptors, those receptors do other things, right? So you can have other problems. So for instance, in fenfluramine, some of those other receptors may be responsible for some of the cardiac issues. And so by being specific to the 2C, we may be able to eliminate the need for cardiac monitoring because we're not binding to the receptor that causes that. So this is some data from their early study. This was a basket trial, so there were 52 patients in this that were randomized to therapy, and they had multiple different etiologies. So 4 of them had Dravet syndrome, but they had people with LGS, and they had people with other developmental epileptic encephalopathies. What they showed in this initial study is they had about a 60% median reduction in seizures overall, and as you can see, the Dravet group had about a 75% reduction. So that's really encouraging information. That's in line, similar to fenfluramine. But keep in mind, there's only 4 patients. So there's only 4 patients with Dravet. So it's small, but it gives you the signal that we've got something that we need to probably study more. It was a well-tolerated drug. The side effects you see there, somnolence, decreased appetite, constipation, these are the kinds of side effects we typically see reported in most anti-seizure medication trials. Nothing else that was really stood out. And then they just released some data recently about their open label extension. So these were people who were in that trial who then went on to get the active drug. Basically, everyone enrolled in it, and the majority of them stayed in that trial at 95% of them at 6 months. Again, the median reduction long-term is very similar, 56%. So this is good. This shows you that, that original trial is only, say 4 months. So this is showing you that long-term, the drug is continuing to work. It also shows you that the people who got placebo, who entered this trial, who got the active drug, also did well. They also had 50-some-odd% reduction. EPX 100, which is clemizole, is currently under study by Harmony Biosciences. This is another serotonergic agonist. As you can see there, it binds to multiple serotonin receptors. Put the QR codes on these that are recruiting so that, you know, if you're interested in them, you can scan and get to them and consider them. This is enrolling kids 2 and up, and it is a double-blind, placebo-controlled trial, of which I don't have any data to share with you from that. Soticlestat is a drug from Takeda. This is a novel mechanism of action, works on a cholesterol 24-hydroxylase inhibitor. So basically, you got this cholesterol gets broken up by an enzyme and it becomes this activating, stimulating form of cholesterol. The idea is if you can reduce that activating, stimulating type of cholesterol, maybe you'll improve seizure frequency. The enrollment for this has completed. This was the original phase 2 data. The phase 2 data included people with both Dravet syndrome and Lennox-Gastaut syndrome. In this original study, which was a small population, again, this was a phase 2, so it's a small population with the disease, they did show in the Dravet for convulsive seizures, you had about a 30-some-odd% reduction versus a slight increase in seizures in the people that had placebo. So that gave them a good sign that maybe they had something worth studying. So they went on and they did a phase 3 trial, which is completed, and the top-line results were released earlier this week. And unfortunately, the primary endpoint, which was the reduction in convulsive seizures during treatment compared to baseline, did not reach significance. So unfortunately, they did not reach what was the primary outcome. They had some secondary endpoints that did, but because they didn't reach the primary endpoint, there'll be some discussion with the FDA as to what they do with this program moving forward, whether they try to move forward with this drug for Dravet syndrome or not. A couple drugs that are already out on the market and available have some studies that are really just looking at a younger age group. So if fenfluramine is approved for ages 2 and up, this study is looking at kids 1 to less than 2 years. That one is recruiting. Jazz has a study of CBD, their Epidiolex. Again, same thing, studying it in age 1 to 2 years. So those are also recruiting. Okay, now finally, disease-modifying therapies. We're almost done. You were 1 step closer to quesadillas, okay? All right. Antisense oligonucleotide therapy. This is STK-001. This is the first of our disease-modifying drugs that we're investigating. I am going to give you the simple guy from Mississippi explanation about how this works, okay? So everybody's walking around with 2 copies of SC and 1A. If you got Dravet syndrome, one of your copies not so good, okay? And so you only got about 50% of good stuff, all right? In general, your DNA gets made into messenger RNA. Messenger RNA gets read by a ribosome, gets made into the protein, which is the SCN1A channel. When that messenger RNA is being made, sometimes it has something in it called nonsense-mediated decay exon. This is a little piece of material that when the ribosome's reading the RNA and it gets to it, it says, oh, throw this out. We don't need this copy. Right? The reason for that is we don't need too much of anything, and so the body naturally throws away some otherwise healthy stuff that it doesn't need. But if you got Dravet syndrome, you need all the healthy stuff you can get, right? And so this is a drug that's given, it's given by spinal tap, lumbar puncture. We give the ASO. The ASO goes and it binds to that nonsense-mediated decay exon. And the reason that works is because in order for messenger RNA to be read by the ribosome, it needs to be single-stranded. When that ASO binds to it, it now becomes double-stranded. And so the ribosome can't read it anymore. If the ribosome doesn't read it anymore, now it's not going to destroy it, and instead it's going to make it. And so what happens in this approach is that we basically capitalize on the good copy of the gene, and we make everything we can make from the good gene to replace what's not being made by the unhealthy gene. So this has some results, which is great. So we had the Monarch trial in the United States. In the Monarch trial, we used multiple different doses because, again, this was a trial to establish safety, but also to figure out which dose is the dose that might work. So they used 10 milligrams, I believe 10, 30, and 45, and then ultimately 70 in the United States. And then in the UK, they used multiple doses as well, with more people getting 70 milligrams. And we did both single doses and we studied multiple doses in that trial. And basically, this is kind of an overview of that data. That green line, that's the 70 milligram dosing, folks. So as you can see, 70 milligrams did much better than people that got 30 milligrams, did better than people that got 45 milligrams. And then when you look at 3 months after the last dose or 6 months after the last dose of this drug, the pink there is the 70 milligram dose. So it had about an 80% reduction of seizures, almost 90% at 6 months in the 70 milligram group versus 45 versus 0 to 25% basically in the lowest dose group. The next thing they looked at is whether you got 1 dose of 70 milligrams. So it's clear 70 milligrams seems to be better, good. Now, is 1 dose or multiple doses better? And if you look at that, 1 dose works, you get a good reduction. However, multiple doses, 2 or 3 doses, had even better overall seizure reduction. So from that, we can take a good feeling that 70 milligrams is the dose we like, and probably multiple doses of 70 milligrams is a good way to start treatment with this. Then we have an open label extension. People in the open label extension would get doses of 30 or 45 milligrams, and basically what we've seen so far is that those doses maintain the response. So if we give 70 milligrams, a couple doses of that, get you some good seizure reduction, then we believe we could give like 45 milligrams and kind of maintain that over time. Obviously, that remains to be completely studied, but that's the thought process behind this. But it's a disease-modifying drug, so we got to show it does something other than reduce seizures by a lot. So again, some of you may have participated in the Butterfly Natural History Trial. That was a study where basically we were doing lots of these developmental, intellectual, cognitive scales to understand the natural course of the comorbid issues in this condition. That told us what the natural history was, and so that's what's in blue, and then that's compared to what happened to the people that got STK001. And so this is the information from the Vineland, which showed that there were improvements over what would have been what happened in natural history in both expressive and receptive communication. The brief, which I talked about earlier, which is a scale of executive functioning, like how you organize thoughts and memory, showed improvement. So on the brief, negative numbers are better. So we have more negative numbers with treatment than you saw without treatment in the natural history. And then this is the global impression of change, which also showed over time compared to the natural history that both the caregivers and the clinicians felt that people were much more likely to be improved or very much improved compared to what we would see in the natural history. Overall, this treatment was tolerated quite well. 30% had some treatment-emergent adverse effects, of which CSF protein elevation and procedural vomiting were most common. There was a single patient who had a SUSAR, which is a sudden unexpected adverse event. And then with the CSF protein elevation, so 75% of the patients had a CSF protein elevation. None of them manifest any symptoms from that, and only a single patient dropped out of the study because of the protein elevation, even though they were asymptomatic. And then the final one I'll talk about is the one that we're just getting started on. That's ETX-101 from Encoded. And so this one's a little different. So the ASO therapy that I just showed you, that's given by spinal tap. ASOs are something that get broken down over time, so they need to be redosed. And so just on average for that study, it's like every 4 months you get a new dose. And they work on the RNA. This is an attenuated adenoviral therapy that's going to work on DNA. And so what we would believe is going to be the difference here is that when you alter the DNA, you're done. It's a one-time thing. And so what they've created here is you have an attenuated adenovirus. So you've got a virus. Adenoviruses are common. Just like you can get a virus that maybe infects your lungs, or you get a virus that causes you to have GI issues, you can create a virus to infect very certain things. So what they've done here is they've created a virus that infects basically the inhibitory GABA neurons that are the problem in Dravet syndrome. So this is going to be a treatment that's given intraventricularly, meaning we're going to drill a hole in the skull and we're going to put it in and we're going to drop these viruses off into the ventricle. That virus is then going to basically go infect the inhibitory neurons. When it infects the inhibitory neurons, it's going to drop off its infection, but its infection is a transcription factor. So it is a piece of material that's going to bind to the DNA and it's going to tell the good, healthy DNA, make more, make more, make more. And so what happens is it goes in, just like the ASO, we're capitalizing on the healthy version, and we're going to make more of the healthy SCN1As to make up for those that are not healthy. So this is just preclinical kind of information right here, but these are like mice models. And so in the first one here, you see that the line at the top basically shows you that the mice that got this therapy survived, whereas mice with Dravet syndrome typically will die early. The second, the HTS assay, is a heat assay. So basically, how much heat can they tolerate before they begin to have seizures? And again, those that got ETX 101 could tolerate much more heat without having seizures than those who had wild-type Dravet. So this study is being conducted in Europe, in Australia, and in the United States. In the United States, it's called the Endeavor trial. There are 2 sites currently. One is Cook Children's, the other will be UCSF. These are very small trials. Originally only 4, that number of patients may grow, and we are enrolling people aged 6 months to 3 years in that trial. So, now, the conclusions are, first of all, clinical trials are absolutely essential to developing new treatments. Like, we should be really, really happy that people are interested in Dravet syndrome and people are trying to develop therapies for Dravet syndrome. And I know we're all tired probably of doing clinical trials, but we don't have a cure yet, do we? So we need to keep working on it. So I think it's a great thing for people to think about participating in. It is a commitment. It's a commitment for you. It's a commitment for me. It's a commitment for everyone involved, but it's worth it. There are resources available so that you guys can identify trials that you might be eligible for. Dravet actually has their own website, dravetclinicaltrials.org, that you can go to and see what's open and recruiting. And lastly, I ended with hopefully what gives people hope. We've got a lot of therapies and new therapies being investigated, and I'm pretty excited about the potential of them. So that's great. And with that, go enjoy a quesadilla.